Thank you for the opportunity to share some of the work that we're doing in my lab. I'm going to talk today about some vaccine efforts that we have going on to target Chlamydia trachomatis adhesion factors and also proteins involved in pathogenesis. So I just wanted to start by showing the picture of my lab and talk about the individuals who are involved. I'm Kathryn Frietze. I'm a principal investigator, and I am an assistant professor in the Department of Molecular Genetics and Microbiology. Also working on this project is an MD Ph.D. student, Amanda Collar, a technician, another Ph.D. student, Anzoa Jamous, and then also an MD student here at UNM. And we've had funding from a number of different grants from the NIH to support this project. So just to start out and talk a little bit about Chlamydia trachomatis, it's a global health problem. Chlamydia trachomatis causes blindness. It's one of the leading causes of infectious blindness. And this is primarily a problem in low and middle income countries and developing nations where there's not very good sanitation. Chlamydia trachomatis is also a sexually transmitted infection. It causes infertility and pelvic inflammatory disease in women. It causes eye infection in babies as they are born to women who have urogenital Chlamydia trachomatis infection, and then also causes urethritis in men. Globally, this map is showing the number of infections. This is even higher now. This is an old map. And you can see that trachoma, which is caused by Chlamydia trachomatis, is really a problem in sub-Saharan Africa and also some in Latin America and the Middle East and Asia. Whereas urogenital infection, sexually transmitted Chlamydia trachomatis, is a problem worldwide. A little bit about Chlamydia trachomatis itself. It's an intracellular bacterium. It actually exists as two different forms. The first form is what's called an elementary body, and this is the infectious form. So when an elementary body attaches to a host cell, it enters and then differentiates into what's called a reticulate body, which is a metabolically active form of chlamydia. It establishes this really large vacuole inside of the infected cell, and that vacuole is called an inclusion. It creates a nice home for the chlamydia to replicate. And I'm showing here a fluorescence microscopy image that we obtained from our lab, showing this large vacuole here in red and green. And then it's almost as large as the nucleus of the cell itself. After more elementary bodies are made in this inclusion, it then goes on to exit the cell where the elementary bodies are released and then can go on to infect other cells as well as other people. And so we're really interested in developing vaccines that target primarily this attachment and adhesion step. If you can block this step, then you can really block infection itself and prevent Chlamydia trachomatis infection as well as the downstream effects of chlamydia. So what our approach is to use bacteriophage virus-like particles, which Bryce did a nice job of introducing in the last talk. And then we've identified a number of chlamydia protein targets. Those are listed here. MOMP, PMPD, PMPC, PGP3, and OMCB. Most of these are adhesion factors that have been shown to be really important in that attachment and adhesion step that initiates infection of chlamydia. From those protein targets, we have identified a number of short peptides that we're interested in targeting. These are peptides that have been shown to be really either immunogenic in humans or in non-human primates, or we know that these peptides are involved somehow in the functions of the chlamydia proteins. Once we have these peptides, we attach them to the surface of a virus-like particle, which allows them to be highly immunogenic. And then the idea is to immunize people and then provide protection from Chlamydia trachomatis, both urogenital infection and also the ocular infection, which causes blindness. So how is this different than other vaccine strategies for Chlamydia trachomatis? So there's been a number of attempts to make Chlamydia trachomatis vaccines in the past, going back 70 years. And currently there are no licensed vaccines for chlamydia. One thing that makes our vaccines unique is that we can easily make them into mixed formulations, which allow us to target many different chlamydia proteins, which are involved in adhesion, but also target many different chlamydia types. There are differences between these proteins among the different chlamydia types that are out there. We also plan to target many adhesion factors, as I mentioned, which can provide better protection against infection than just targeting one protein or peptide by itself. And also something that I didn't mention yet is that the result of chlamydia infection, so the pathology that comes from chlamydia infection, is really thought to be driven by T cells. And so because of this, vaccines that elicit T cell responses could lead to actually bad results down the road. And so our vaccines, we think, are a better approach than other vaccine approaches because we really only elicit antibodies to chlamydia with our approach. And so we think that our approach is likely to be safer than some of the other approaches that, that groups have taken for chlamydia vaccines. In terms of the need for chlamydia vaccines and the market potential, the World Health Organization and the NIH have identified Chlamydia trachomatis as a target for vaccine development. And just some statistics, it's estimated that 1 in 5 people at any given time in the United States have a sexually transmitted infection, and chlamydia is one of the most common. Every year that's actually going up. We are seeing a rise in Chlamydia trachomatis infections that are sexually transmitted. And this is resulting in really high medical costs in our country and also around the world. In terms of the market for the vaccine, we would anticipate that this would include women who are at risk for chlamydia, trachomatis, urogenital infections, and this would be a similar market to the HPV vaccine Gardasil. But we also think that our vaccine would include children who are at risk for ocular trachoma globally. Just to share some data and results that we have so far from our chlamydia vaccines, I'm showing you here an ELISA. So this is showing antibodies binding to actual chlamydia trachomatis elementary bodies from different types of chlamydia. And so what we did is we had a mixed vaccine targeting several different peptides. We immunized rabbits with this. And then the sera, or the blood from those rabbits, we used to see if those rabbits had antibodies that could bind to actual chlamydia. And we see indeed that all of the chlamydia types that we tested in this assay, we do see binding with the sera from our vaccinated animals. Here, I'm showing you that our mixed vaccine elicits high titer and long-lasting antibody responses in both the blood and the urogenital tract. So here, this is, again, ELISA is looking at binding of antibody. This is actually to one of the peptides that, that was used and was a target with our vaccine. We see that we get high titer antibodies, and those seem to last for up to at least 24 weeks. We're continuing to look at later time points in the mouse. And then we also did vaginal washes where we look at antibody at the actual site of infection for urogenital infection. And we do see antibody responses in the vaginal tract as well. Finally, we've done a challenge study with a mouse model of chlamydia And here we see, so we have a version of chlamydia that expresses luciferase, which allows us to look at the infection in the mice using what's called an IVUS. And so here, these are mice that were immunized with our vaccine. These are control mice in the bottom row here, and we can see a pretty substantial decrease in infection happening when we have our vaccinated animals compared to control. So this was really promising data, and we're continuing to follow up on this with additional challenge studies to test our vaccines. So in terms of our current status, we've developed a number of vaccine candidates against the 5 different chlamydia proteins that I showed earlier. We've shown that they are immunogenic, and we've also looked at longevity of the vaccines. Those studies are ongoing, but we expect that they will, as Bryce showed earlier, have really long-lasting antibody responses, which is great for a vaccine. And as I showed, we've begun challenge studies to look at the efficacy of our individual vaccine candidates. The next step then is to identify the most promising individual vaccines, mix them together in one formulation, and then test them in mouse challenge models. In terms of funding that we're pursuing, we have 2 grants right now that we're pursuing to get additional funding to support this. We have an R21 that was just submitted to the NIH, and then an R01 that was previously submitted and received a score, and we will resubmit that in February 2022. Thank you for listening, and this is my contact information here.